Welcome to the podcast of Hemisphere, the official journal of the European Hematology Association. Hemisphere's podcast presents insightful, expert discussions about recent hematology publications. We hope you enjoy.
Hello and welcome to the Hemisphere podcast. My name is Charles de Bock and I'm your host. p53 is one of the most frequently mutated genes in solid cancers. It's a critical hub for fate decisions linked to DNA damage and metabolic deprivation. It is also a transcription factor and is, of course, described as the guardian of the genome. When I actually searched for p53 in cancer in PubMed, I had over 95,000 articles. But if I did the same for p53 in myeloproliferative neoplasms, I kind of found less than 100 papers. And even though the presence of p53 mutations are associated with very poor overall survival for those patients, there's not that much literature. So why is that? And what is the role of p53 in normal hematopoiesis, as well as hematopoietic malignancies, especially myeloproliferative neoplasms? Well, to learn more, I am excited to be joined by Dr. Isabelle Plo from the INSERM Gustave Roussy University Paris in Saclay, France. I know I didn't say that correct, but that's okay. And then Dr. Barbara Monte-Mór from the Instituto Nacional de Câncer in Rio de Janeiro in Brazil. So we have someone in Europe, someone in Brazil, and then myself in Sydney. So we've almost got all the corners of the globe. And they are the co-authors of a very new and timely review, now published in Hemisphere, called Unraveling the Role of TP53 Mutations in Myeloproliferative Neoplasms, Molecular Mechanisms of Leukemic Transformation. Welcome to you both.
Thank you very
I'm
much.
going to start off with,
Thank
it's great that you're both here and the time zones, we've worked out the time zones, which is super exciting. Okay, so question one, I'm going to launch in it. We should really start with the role of p53 in normal hematopoiesis. And so what do we know about p53 and its role in hematopoietic stem cells?
So maybe I can start to try to answer this question. When we think about TP53, we think about it as a tumor suppressor. But really in hematopoiesis, it has a more broader roles, particularly in controlling the hematopoietic stem cell biology and also the lineage differentiation. And most of what we know about the TP53 role in hematopoiesis comes from the TP53 knockout models. These mice are viable, but their hematopoiesis are profoundly modified. So if we start with hematopoietic stem cells or HSCs, in these TP53 knockout models, generally HSC are increased in numbers. And this is because p53 enforce the quiescence and let the cells in resting state through activation of GFI1 and Nectin. So in the p53 knockout model, generally the HSC cycle and expand, and generally also transplantation assay using the TP53 knockout bone marrow cells outcompete with the normal cells in the short term, but they are impaired because in the long term they fail to reconstitute the normal hematopoiesis. So it means that p53 has also a role in maintaining the quiescence and the long-term activity of p53. It's not just controlling the proliferation. Now, if we take what is its role in the lineage differentiation, and we start with the erythropoiesis, it seems to control, in fact, the proliferation and differentiation of this lineage. In fact, under steady state, the TP53 knockout mouse model are nearly normal, but under stress conditions, they recapitulate the hematopoiesis faster than the normal mice because of the increased erythroblastic proliferation. So this is important also to mention that p53 is transiently activated during the erythroblastic differentiation under the ribosomal stress. In fact, in this case, it controls the proper differentiation of the red cells, and also it it eliminates the abnormal erythroblastic that are not good for the proper differentiation. Now, in the megakaryopoiesis, again, p53 is a brake, and its loss in p53 knockout mice increased the megakaryopoiesis, also increased the hyperploid megakaryocyte. And so in this context, also the cell generated are abnormal and the platelet generated are impaired in function. So overall, p53 play a real key balancing role in hematopoiesis, ensuring long-term and quiescence in hematopoietic stem cell, but induce also a control in the lineage differentiation, particularly erythroblastic and megakaryocytic differentiation. And so too little p53 leads to expansion, and too much p53 leads to toxicity. And this balance is exactly why disruption of the TP53 pathway is very important in hematopoiesis and is important for the development of hematological malignancies.
It's super interesting. So you really need this like Goldilocks level of p53 expression. Not too much, but not enough. And it's very highly regulated. I mean, I find it fascinating that the loss of p53 actually causes this increase in stem cell fitness, but only transiently before they exhaust over the long term. I find that fascinating. Barbara, did you have any other thing to add? Otherwise, I'm going to keep going.
Yes, you can keep going.
Okay. So your review then moves to myeloproliferative neoplasms, and that includes essential thrombocytopenia, or ET, and polycythemia vera, or PV, as well as primary myelofibrosis. I know from the literature that the most common mutation in these cases is often the JAK2 V617F mutation, which is an activating kinase mutation. So how does the p53 mutation sit in the landscape of that mutation, and what's known about the relationship between the JAK2 V617F mutation and p53 perhaps?
Maybe I'll answer this one first. So indeed, as you mentioned, the JAK2 V617F mutation is the most common mutation in myeloproliferative neoplasms. It's a mutation that arises at the hematopoietic stem cell level, and it leads to constitutive activation of the JAK-STAT pathway. So this ultimately drives the overproduction of red cells that we observe in these diseases. So what's particularly interesting is that this mutation doesn't only promote proliferation of these cells, but they also seem to downregulate p53 function. So the group of Dr. Isabelle Plo and also myself have working on these diseases, and we have for quite some time now shown that JAK2 V617F mutations increases MDM2 expression. That's a key negative regulator of p53, and so by this way, this increases p53 degradation. There is also another layer of regulation that's being described. It's a protein called PPIL2 that's a STAT5 target, and it's also seen to be upregulated in MPNs, and it acts also as a 3 ligase for p53, enhancing its degradation. So overall, in chronic phase MPN, what we have is this kind of environment while p53 activity is being downregulated at multiple levels by the signaling itself. In terms of mutation, TP53 mutations are quite uncommon in the chronic phase. What we saw in the literature was about 7% of MPN patients mutated in chronic phase. However, this frequency of mutations increased significantly during leukemic transformation. In secondary acute leukemia, we can see roughly 1/3 of patients carrying one or more TP53 alterations. And it's important maybe to mention that these alterations include not only point mutations, but also other kind of chromosomal alterations involving, for example, chromosome 17p, where the p53 locus is located, and as well as other genetic alterations that alter key negative regulators of p53, such as MDM4 or PPM1D. So altogether, this means that the genetic disruption of p53 pathway in multiple genetic events is a relevant mechanism for the leukemic transformation in these diseases. And maybe we can also add that in secondary acute leukemia, we not only see that these mutations are more frequent, but we also see that the clonal burden increases. So the variant allelic frequency is very high at the leukemic phase, and it often exceeds the 50% threshold, which maybe is consistent with the loss of the Y-type allele, and that is a very important event. In fact, in acute myeloid leukemia secondary to MPN, we see that very frequently the patients have more than one alteration in both TP53 alleles, which we call multi-hit status. What we can summarize is that in chronic phase, these mutations are uncommon, not very frequent, but present at a low variant allelic frequency, and they can remain like this for years. But once we have the two event hit and loss of the Y-type allele, so this B allelic inactivation, and this become a key trigger to leukemic transformation to progress into acute leukemia.
That's the ultimate question, I think, as well, in terms of, how does that change from what is that trigger and how come you get this chronic phase and then the ultimate acute frank leukemia, which generates. But if I can take you back to the types of mutations in p53, because from reading your review, not all of them are the same. And I always think, mutation means inactivation, but actually that's not the case. And, you can get deletion of p53, which means complete loss of function. But I was intrigued that you have this missense mutation, which is at you corrected me before, R172H, which is in the mouse, which is equivalent to the R75H in the human. So what's the difference between complete deletion and this particular point mutation? Maybe you can describe that for me and the importance of those.
Well, that seems to be a really important question because indeed not all TP53 mutations or alteration behave the same way. If you take, for example, deletions or truncating mutations, that will essentially eliminate the or suppressor function of TP53. The cell will lose the ability to respond to common stress, for example, DNA damage, and it's an important barrier to malignant progression that will be lost in this cell. However, for p53, more than 80% of the mutations are indeed missense mutations. And the consequences of missense mutations, particularly these, what we call hotspot mutations that are located in the DNA binding domain, this can be a little bit more complex. So as I mentioned, they typically will happen in the DBD, the DNA binding domain, and it will impair the ability to regulate canonical targets of this transcription factor. And this is kind of loss of function. But beyond this loss of function, these mutant proteins seem also to be relatively well expressed and stable, and they have been described to exert other functions. So, in particular, there's been dominant negative effect that has been also investigated mainly in solid tumors, but also in leukemia. And in addition, there is evidence from solid tumors and from experimental models that mutations such as the R175H mutation can acquire new functions, what we call oncogenic activity. And this occurs by many mechanisms, regulating different proteins or regulating other target genes.
Yeah.
In AML, it's more or less controversial because there has been evidence in, for example, cell lines carrying these mutations that show more of a dominant negative effect. And on the other side, there are evidence from murine models. Maybe, I don't know, we'll go through murine models later that support more an oncogenic gain of function, particular induction of certain mediators that can activate new pathways in these cells, and this will lead to new oncogenic functions in the cell.
Yep.
So it's quite different from loss of function.
Exactly. I like the description, which was, if you lose p53, you're removing the brakes. But if you have this point mutation, you're not only removing the brakes, but you're also putting the accelerator on. And so that's sometimes where this dominant negative effect is from the stability of the protein, which is super exciting. You mentioned the mouse models before in terms of our understanding of normal hematopoiesis, and your review does a really nice job of describing some of the different mouse models that have been looked at in terms of understanding the relationship between JAK2 mutations and p53, in particular, really understanding, the cell of origin and how they transform cells into acute leukemia. Can you give us a brief description on some of the models which have been really useful and how they've helped us understand myeloproliferative neoplasms with respect to p53 mutations?
So maybe I can try to answer this question. Mouse models have been very important, in fact, to understand the cooperation between JAK2 V617F and TP53 loss. And in fact, they have shown us that both the genetics and the context matters. There have been a lot of mouse models that have been generated. First of all, that were the retroviral bone marrow mouse model. In this type of model, you overexpress the JAK2 V617F in TP53 knockout bone marrow cells and transplant these cells into lethally irradiated mice. And in these mice, the picture is very clear. First, these mice develop a rapid MPN-like phenotype, and then they rapidly progress to very aggressive erythroleukemia. Also, what was very important with this model is that it identified the key cell driver of the disease, which are the megakaryocyte erythroblastic progenitor, or MEPs, that present a lot of features of genetic instability and also show, very interestingly, a strong dependence to JAK-STAT signaling pathway, which is important then for therapy also. But these models are not perfect because in this case, you overexpress JAK2 and you have a p53 complete loss. So it does not model completely what is found in patient. And that's why the knock-in mouse model come in the game. So in this model, you express the JAK2 V617F and the TP53 mutation at physiological levels. And again, in this model, the picture is very clear. Monoallelic mutation of TP53 is not sufficient to induce the leukemia. You need the biallelic alteration of the TP53 to trigger the leukemia. So this model closely matches what is seen in patients because in patients that develop leukemia, they all present multi-hit TP53 cells. So indicating that really TP53 complete loss is a crucial turning point to trigger the disease. Also, what was very important with this model is that we observed that most of the transplantation were more rapid in a transplantation experiment in which you have a huge inflammatory and DNA damaging context. So this could suggest that stress is very important also to enhance the leukemia. And supporting this, there have been some experiments recently described in the paper in Nature Genetics from our group and the group of Adam Mead, showing that interferon is able to induce in TP53 heterozygous cells karyotypic abnormalities that is associated with genetic instability and linking that with inflammation and the development of leukemia. So altogether, we can say that JAK2 V617F drives the proliferation, TP53 loss removes the control, and stress and inflammation push the system over the edge to trigger the leukemia. So what remains unclear now is how the mutation act. Are they only loss of function or really gain of function, as said Barbara? It's very important to understand all this function of this missense TP53 mutation. And also maybe what is important now is to understand the different steps of the disease and particularly how the biallelic mutation of TP53 occur. This is very important. And potentially in this role, study the role of maybe therapies that were given previously in chronic phase of MPN. Because it's known that TP53 cancer most of the times linked to therapy. So it's very important to try to investigate the effect of this therapy. But all these models have been very essential, not only for understanding the disease pathology, but also start to be used as a platform also to test new therapeutic strategy.
Which I think we're going to touch on right now, because I think the mouse models are amazing. There are lots of them, and I think that they still have a critical role. As you discuss, I'm really intrigued by the inflammation and how that relates to the human position and whether we know much about that. But before I go into that, you hinted on the whole therapy part of it, and you discuss some of that in terms of some exciting developments. In particular, you discuss the role of allogeneic stem cell transplantation, but also how to reactivate p53, but especially in those with non-mutated p53 MPNs. But first of all, maybe we can touch on stem cell transplantation and what are the challenges for those with p53 mutations and MPNs? Like, how effective is stem cell transplantation?
So maybe I can try to discuss this a little bit. So although this allogeneic transplantation is potentially curative for these patients, the role of this therapy in TP53 mutated patients, in MPN patients, is quite limited. So part of the difficulties are related to patient itself. So they often present advanced age and comorbidities that can restrict the application of allogeneic transplantation. But there is also a major issue that is underlying the disease biology, as we have been talking about. The fact that the cells are TP53 mutated and often are also B-allelic mutated really changes the biology of this disease. So there are not so many studies on allotransplantation in this population, but recent studies show that the outcome of these patients are significantly worse in the mutated context for myelofibrosis especially, and also especially for patients with this multi-hit status. So there is a very high rate of post-transplant relapse in this patient, which often exceeds 50% in the subgroup of multi-hit mutated patients. And there's also recent evidence suggesting that maybe these patients could benefit from earlier transplantation. So in secondary acute myeloid leukemia, this is also a very drastic situation. And it's maybe even more challenging. There's very low survival rates for these patients reported with TP53 mutated patients. So overall, this remains an option, but it certainly needs to we need to move on to try to find new strategies that will target this pathway in the disease. So reducing the relapse risk and potentially earlier intervention for these patients.
Yep. I mean, you do discuss that as well in terms of perhaps restoring p53 function using MDM2 inhibitors, at least in non-mutated p53 cases, is one potential strategy. However, I did note that you showed that clinical trials show that when you do this, you can inadvertently cause the expansion of pre-existing minor clones which have the p53 mutation. So, that's obviously concerning given the fact that it's so dire and then you have limited options. What are some of the exciting developments that you see coming through the pipeline, at least to try and combat this and treatment of maybe the more, as you said, acute cases of mutant p53 MPNs?
There is a lot of new therapeutic strategy in this field. It's moving very fast. But really targeting p53 in MPN is a double-edged sword, because in classical myeloproliferative, as you said, in which TP53 is not often mutated, MDM2 inhibitor has been used to antagonize the interaction between MTM2 and p53 and to restore its stability. And this makes sense because, as Barbara said, JAK2 V617F leads to p53 suppression through an increase in MDM2 protein. And in clinical trials, although it is very useful for symptoms relief and also for hematological parameters. Also, it decreased the variant allele frequency. That is very important because it can show that it can long-term modify the disease, the history of the disease. But as you said, that there are some limiting effects because it induces a lot of toxicity, mainly the gastrointestinal cytotoxicity. And more importantly, there is in some patients where there are some p53 mutated pre-existing cells that are selected by the therapy. So hopefully after cessation of the therapy, they disappeared, but this is a limiting effect in such patient that carries this TP53 clone. So in parallel, the field has moved quite quickly now, and the problem is how to fix this mutant p53. So one key approach is the p53 reactivator. In fact, these type of molecules are able to reconform the mutant p53 into a p53 normal conformation. So it's fantastic. And the most advanced compound is the APR246 or eprenatopop. I don't know if I pronounce well. But this is one of the most advanced that have been used not in myeloproliferative neoplasm, but in acute myeloid leukemia and myelodysplasia with very encouraging results in phase 1 clinical trial. But it seems that in phase 3, it was a little bit disappointing. And so this drug now is combined with other inhibitors, such as the venetoclax, that is a BCL2 inhibitor. Another compound reactivator molecule is the resatapop. This one is more specific to the Y220C mutation of TP53. And so it restores the proper protein folding. And this is clearly a more specific drug, but it has also started some clinical trial with this compound in myelodysplasia and acute myeloid leukemia. So beyond this reactivation of p53 in mutant case, there are another strategy that are more based on trying to use the vulnerabilities induced by TP53 mutation because it induces DNA damaging context. So these cells are very sensitive to inhibitors such as PARP or WEE inhibitor that plays a role in DNA damage response. You can try to use the vulnerability in the metabolism also and try to inhibit some anti-apoptotic molecules such as BCL-XL. There are also more specific approaches, for instance, targeting the mutant CALR cells. In fact, in our lab, we have recently shown that monoclonal antibodies that are directed against mutant CALR are able to target the CALR mutant cells that are also TP53 multi-hit in a xenograft model. So this is very important because there you can see that is a strong dependence of the TP53 mutant cell to the JAK-STAT signaling pathway, because with this antibody you inhibit the JAK-STAT signaling pathway. So maybe it could be also an alternative strategy, but that is interesting. And finally, there are some really emerging strategy and novel strategy that are based on gene editing, oncolytic viruses, and also exploiting some neoantigens that are specifically expressed in TP53 mutant cells, and so generating immunotherapies in such case. So the field is shifting toward precision medicine in TP53 mutant case, and trying to restore proper folding of TP53 or targeting some vulnerabilities. And this opens new avenues for the high-risk MPN that are a little bit less studied than AML or myelodysplasia at this point.
Yeah. I'm always astounded. I'm so glad there are some very clever chemists who can work out how to have compounds which can target essentially a transcription factor which is often seen as undruggable. These aren't kinases with ATP binding pockets where you can make beautiful drugs, that they can design drugs which can do refolding, which is very exciting. I didn't even realize that they were out there. I do like the fact that we can exploit the vulnerabilities. It reminds me a lot of the BRCA1 and breast cancer and olaparib with the PARP inhibition as well, the same, synergistic impact, which is fantastic. So I think that's that the future is bright. Although, as you said, we have to probably combine them with different drugs. I noticed you said BCL-XL, which is, if memory serves, targeted by navitoclax, but I think navitoclax is just discontinued. That's a bit of a disappointing avenue for that particular drug. So perhaps there will be other BCL-XL inhibitors coming through.
Maybe it's discontinued. Maybe it's discontinued in high-risk MPN, but maybe in post-MPN leukemia could be also another alternative to use this.
Yep, true. Well, writing a review is always a challenge. And, I could keep going on in your paper, but I really encourage the listeners to read your paper. It's really well written. But on the writing topic, as I mentioned, I'm in Sydney, but you're both on different sides of the globe, and you wrote this beautiful review. How did you go about writing it? What advice can you give to prospective authors who are thinking about writing their own review in terms of how to limit the scope? Because, and I did mention p53 in cancer is 100,000 papers. Okay, you might have 100 papers, but sometimes having fewer papers is more challenging to write a review. Can you give some advice, and how was your journey when you wrote the review together?
Maybe I can tell a little bit how it was for me, and then you can maybe add some information. So indeed, it was really a lot of work. For this review in particular, the idea of writing it and also the scope of what we were discussing, that came quite naturally because we were indeed working on projects, both our group in France, in Brazil, working on p53 in MPNs. So that was really advantage because we are already dedicated to the literature and so on. So the group of Isabelle in Villejuif has been working on p53 in MPN for quite some time now. She maybe comment more on that. The scope of the review really came from the expertise in the group, among our co-authors, and also common interest in the field. That part was okay. But then come the fact that, as we mentioned through our discussion here, the field is moving quite quickly. So there are a lot of new studies on multi-hit status, a lot of new studies on new drugs. So actually, while we were writing, there are always new literature coming.
True.
So we are always trying to fit some new articles that just came. This actually made a lot of back and forth between France and Brazil. So we have to Zoom meetings sometimes to discuss new papers. It was quite an adventure, but it was very interesting indeed.
Excellent. Isabelle, your take on writing a review?
Yes, so from a personal point of view, we started to work on p53 20 years ago, and to try to understand its role in hematopoiesis and particularly in myeloproliferative neoplasm. And at this time, the field was not so convinced on p53. They thought it was no role, it was only for solid cancer. And because very few people were mutated on TP53 at that time. So since then, we have followed very closely the literature, and we have expanded also our project mainly now on calreticulin mutation and TP53 cooperation, particularly with the group of Barbara in Brazil. We exchange some students and we have common project. So really for this review, it was the expertise that motivates us. And also, we don't try to be exhaustive, but try to be more coherent and to try to write more what is known, what is debated, and also where the field moves. So finally, it was a hard job because it takes us a lot of time. Because as Barbara said, the field is moving very fast, and we had to add a lot of reference each time. But I hope this review will be inspiring and insightful and timely to other people, maybe to have new hypotheses on TP53 to be able to understand better the field.
Excellent. I think, what makes it really nice is that the review is more than a collection of facts and papers, because I could find those papers, but you've distilled it in a nice narrative. You've made it understandable. And I really like how you have really put your kind of an opinion on what you think is the direction of the field and where the gaps are in the field. I think that's really essential, because then it helps us understand where we should be going on our research. Also, it's hard to believe that, into 20 years, how the field for you has moved, because you've said not many people thought p53 was important. And just that's testament to perseverance and that grit of having to follow up with a project where you really believe that, no, this is important. It's not so much the frequency. And then with time, obviously, that's shown to be true.
Many
so congratulations.
Yes,
Sorry,
many people said, no, drop this project. It's not important. So I said, no, I want to continue. I thought it has a major role in myeloproliferative neoplasm.
I think that speaks volumes. That speaks volumes. I think that's such an important lesson as well, that sometimes don't always listen to others who think what something's important. We often are the best judges of that. So that's fantastic. Okay. Thank you, Isabelle. Thank you, Barbara, for a wonderful chat about your review. I would love to point our listeners to the fantastic manuscript, which is now published in Hemisphere. It's wonderful. As you said, the field is moving fast, and I think you've done a fantastic job in collating those ideas, especially the new drugs. I really like that section in particular. So thank you once again, and I wish you well on your ongoing studies on p53 mutations and MPNs. Thank you both.
Thank you very much.
Thank you for
Barbara.
having. Thank you also, Isa, and thank you for having us in the podcast.
Thank you for listening to this episode of the Hemisphere podcast. All of Hemisphere's content is open access and can be found at www.hemispherejournal.com. We hope you will join us for future podcast episodes.